There we go. I
Perfect.
think it's going to be
Okay,
doing fine. That's all right.
amazing. So sorry for all that faffing at the start there, guys. Yeah, so like I was saying, today we're going to be looking at metabolism, and metabolism's a big topic on the syllabus, but we're focusing in on lipids, glucose, and amino acids today. So let's have a look at my introduction. So, yeah, the content will be focused on the left-hand side of the syllabus for the most part, just because it's so big, which we'll get to in a second. And the content, some of it's adapted from previous year's slides, and I've tried to map it to the following traffic light system where possible. There's three interactive multiple choice check-in slides. And I think the previous from previous sessions that you guys have had is to slow down and to include essay related things. So what I've done, because I'm conscious that we might not get through all of the left-hand side topic just because of how big the topic in general is, I've put loads of clinical bits and experimental evidence at the end of the slides so that if we have time, we can start to have a look through them, but you'll also get a copy of the slides so you can have those notes just in general. And on that note as well, a lot of the slides are quite wordy, because I thought you guys might want to use them and come back to use them as notes, but we'll see how we get on. Anyway, the tutorial essays I did in first year for metabolism are all listed here as well. And if you want them, please just email me for access to those. I'm happy to share them. And most of them have feedback on them. I was at a conference this weekend, and one of the sort of poster presentations was about social capital between medical students and how people who share their essays and receive essays from other people and resources are more likely to do well. So I'm more than happy to share things with you. Just hit me up via email, which is on the final slide with the QR code for feedback. And then generally, previous essay titles, I had a look back to when I was in your year, and my year at Keble was slightly neurotic. So we went back through all the different examination reports and found out all the different essay titles relating to each topic. And these are the metabolism ones for the 8 years leading up to 2022 when I sat my exams. So if you want to know the kind of vibes of essay titles, please feel free to use this resource. Obviously, your guys' exams have changed now a little bit because we had to write three essays, whereas you guys write one. So they might have changed, and I'd recommend looking at the exam reports for the years previous to you as well. Anyway, let's get cracking. So these are the syllabus points. Like I said, there's quite a lot of them to get through. We'll start going through them in order, basically. They're not in order on my screen, I've realized. So I'll do fat as a metabolic fuel first, then glucose, and then amino acid metabolism. And if you guys have any questions at any point, please feel free to put your hand up or post in the chat, and I'll try and answer them. But yeah, let's get going. So, fat as a metabolic fuel. The first bit of the syllabus is just an overview, so advantages, disadvantages, and they're all listed here. So, the advantages of using fat as a metabolic fuel are that it has a high energy density, it's efficient storage, it's abundant and long-term supply, and it's used in other pathways. And basically, the only catch of using fat as a metabolic fuel is that it requires oxygen. So it can only be used aerobically. Yeah, and it's just slow in general. Okay, cool. Again, a bit more of an overview in detail. So, it's a major form of dietary fat. Oh no, the major form of dietary fat, sorry, is triacylglycerol. And I'm sure you all know from A-level biology that tags or glycerol backgrounds. Sorry, glycerol backbones with three esterified fatty acid chains. Okay, cool. And then in terms of essential fatty acids, we've got linoleic acid or omega-6 and alpha linoleic acid, which is omega-3, and they must be obtained from the diet. So you'll see a lot of these sorts of things in metabolism, so essential and non-essential. Is everyone happy with what the difference is? The difference is basically that essential must be obtained from the diet. So that's what we're working with. There's a deficiency here. So deficiency of essential fatty acids, dermatitis, stunted growth, poor wound healing, increased infection risk. So all quite easy things to wrap your head around. And then this is just some information on classification by double bonds of fats, which I'm sure you guys have seen in probably A-level chemistry as well as biology. So yeah, cool. And then lastly, in terms of overview, before we get jumping into the actual sort of processes you guys need to be aware of, is an overview of like a fed versus fasting state table. So you'll probably see these in all the lectures that you have where it lists the tissue and compares the kind of fuel that those tissues use, depending on whether they're in a fed or a fasted state. So I'd be aware of this table, memorize it if you can, but it will also come up in the actual specific processes as well. So don't worry too much if you are not good at memorizing tables like I'm not. Okay, so this is where things get sad because my what they called animations, my animations won't work. That's okay. So we're jumping into the actual processes now. Starting with fat transport around the body. So absorption to circulation. So our dietary fats, they're going to be broken down into smaller bits, so monoglycerides and nephers. They're absorbed by enterocytes, reassembled into tags, and packaged with apolipoproteins into chylomicrons. So there's a little overview diagram here, a handy diagram that I found on the internet, um, that shows the process from the lumen of the intestine, I guess, into the enterocytes, and into the bloodstream, and then the lymph, because that's where chylomicrons end up, is the lymph, and then the systemic circulation to deliver fats. So that's a nice overview diagram there. Following that, we have uptake into adipose tissue. So we have lipoprotein lipase on the capillary walls to hydrolyze those tags back down again into nephers and glycerol. The nephers are then going to enter the adipocytes, where they're going to be re-esterified into tags for storage. And here's a bit of regulation. So insulin stimulates LPL, promoting fat storage. I hope everyone's, I hope that's all making sense to everyone so far. And then just a bit more on fat transport. So in terms of lipid mobilization, so we're thinking about releasing fats from storage now. Tags can later be mobilized when they're needed, but that's going to be inhibited by insulin and high plasma glucose levels. And during this release, ApoB100 is used to form VLDLs instead of chylomicrons, which transport endogenously derived fats. So that confirms that they're no longer dietary in origin. So like tags to your body that something's not dietary in origin anymore. Okay, so during fasting and exercise, we have another enzyme, so hormone sensitive lipase, that breaks down tags again. That's activated by adrenaline and inhibited by insulin this time. So the nephers released are bound to albumin for transport, and glycerol travels to the liver for gluconeogenesis. And you can see the plasma nether levels listed there. So in fed state, they're low. Short fast, you find around 300 to 600 micromoles per litre, and prolonged fast, that goes up to 1,300. So that's our plasma nether levels. And then in the image in the bottom left, you can see the structure of a chylomicron. So, cool. That's everything on fat transport around the body. The next sort of step in the syllabus and also logically is the oxidation of fat. So entry and regulation, first of all. So activation of fatty acids. So fatty acids enter cells and are activated by acyl-CoA synthetase, fatty acyl-CoA in cytosol. And that traps the fatty acid inside the cell for metabolism. So you can see it here. So this is the mitochondria, and I guess where they've placed it on this diagram that I got from online is supposed to represent that this enzyme is in the cytosol. Okay. Up next is the carnitine shuttle for long chain fatty acids. So fatty acyl-CoA can't cross the mitochondrial membrane directly. It needs that shuttle system. So CPT1 in the outer membrane replaces CoA with carnitine. It's transported across the inner membrane via carnitine acyl-carnitine translocase, and then CPT2 on the matrix side reattaches that CoA to form fatty acyl-CoA. And just a note, short and medium fatty acids that are less than 8 carbons don't need carnitine. So here is a zoom in of that carnitine in this diagram. So this is the kind of thing you'd probably put in an essay on fat metabolism. So a zoom into this carnitine shuttle, and you can see it here in the overall bigger picture. So localized to the membranes in this overall diagram of what's happening with fat oxidation. Okay. Last but not least, for entry and regulation, we have regulation by malonyl-CoA. So malonyl-CoA inhibits CPT1. So CPT1, if we inhibit it, is going to prevent beta oxidation during lipid synthesis. And you can kind of see that illustrated out here in the fed state, increase in glycolysis, increase in citrate, increase in ACC, increase in malonyl-CoA, which inhibits fatty acid oxidation and prevents a futile cycle of simultaneous fat synthesis and breakdown. Okay. Um, yeah, so this is the regulation there. Cool. So once we've thought about entry and regulation, we've got beta oxidation. So the location of that is the mitochondrial matrix where we've ended up inevitably after we've entered and regulated. The substrate is long chain saturated fatty acids such as palmitate, and the goal is to generate acetyl-CoA, NADH, and FADH2 to feed into the TCA cycle and electron transport chain. And just a note on that, we won't be covering the TCA cycle or electron transport chain today, just because it comes under a separate bit of the syllabus. But I think there is another session on that coming up, or it might have already been. So hopefully you guys are aware of some of the information to do with that, because it comes up quite a lot in these topics. Cool. So going into that in more detail, we've basically got a 4-step cycle that repeats until the entire fatty acid is cleaved. This is, sadly for you guys, a diagram from one of my essays in first year. But basically those 4 steps are oxidation, hydration, oxidation again, and then thiolysis. So it basically just repeats and repeats and repeats, removing 2 carbons each time until the desired product is formed. And you can see there's kind of the specific enzymes listed there as well. So oxidation, we're using FADH2 in our first step. In the second oxidation, it's NAD plus to NADH and H plus. Hydration, nice and easy, adds water, and then thiolysis or thiolysis is cleavage. So you get acetyl-CoA and a fatty acyl-CoA. So in summary, each cycle removes 2 carbons as acetyl-CoA. Palmitate yields 8 acetyl-CoA, 7 ADH, 7 FADH2, which would be around 106 ATP in total. So that kind of just goes to support what we talked about earlier in terms of the importance of lipid metabolism is that it's very energy dense. So one palmitate yields that much ATP. That's a lot of energy in comparison to glucose, which we'll get to in a second. And yeah, it says here regulated by energy needs and mannose-CoA levels. And yeah, I think that is everything you kind of need to know. So left-hand side of the syllabus wise for fatty acid breakdown and kind of beta oxidation. The next thing to be aware of is fatty acid biosynthesis in the liver. Again, apologies for all the text on the slide and the lack of sort of animations to break that up a bit. But in terms of where and when fatty acid biosynthesis happens, it's in the cytoplasm of the liver and adipose in the fed state, and it's triggered by excess glucose and amino acids. In terms of regulation, insulin activates lipogenesis, upregulating ACC and FAS. And then lipogenesis and beta oxidation are reciprocally regulated to avoid futile cycling. And just to note here on key steps and enzymes. So we've got steps one to do with excess acetyl-CoA, so from glucose and amino acid metabolism, like we talked about, that exits the mitochondria as citrate. Citrate goes into acetyl-CoA in the cytosol via this enzyme here. And then we eventually, once we've passed all of these bits again, we get to step 4, so fatty acid synthase complex, which builds fatty acids. And that's basically the reverse operation of what we've already talked about here. So it's the same sort of steps in reverse. There are some differences, but in terms of memorizing things and learning things for this year, you can save yourself some time with that because it's basically the same thing, just in reverse. Cool. And then thioesterase releases the final fatty acid for further modification. And we can see that overviewed here. So we've got citrate leaving from this kind of excess glucose pyruvate pathway up here and ketone bodies here. So we're getting excess acetyl-CoA being entered into this cycle, and then citrate is going to leave to this fatty acid synthesis pathway, which is in more detail here. And you can kind of see this is my essay diagram again. So it is just the reverse of the diagram previously. Okay. I feel like I'm doing a bad job at going slowly. But our next sort of bit on this is ketogenesis. So when this only happens in fasting and starvation, or in an insulin deficiency. So people with type 1 diabetes might go into ketogenesis. And it occurs when beta oxidation generates excess acetyl-CoA, and that saturates the TCA cycle. So the liver uses excess fatty acids to generate ketone bodies, and it's basically a glucose-sparing fuel for the brain and peripheral tissues. And there's some key ketone bodies there. So beta hydroxybutyrate, that's the most abundant, acetoacetate, and then acetone, which it says here it's non-metabolizable, and it's therefore exhaled. And you can often, if people are in ketogenesis, or fasting, starving, insulin deficiency, and type 1 diabetes, you can often smell the acetone on their breath. But yeah. So in terms of steps and what that looks like, our steps in ketogenesis, we've got 2 acetyl-CoA combining. So you can see that on this diagram at the same time, if I keep it still. So you can see that on the diagram. And then, yeah, you've got acetoacetate formation, and then acetoacetate forms this beta hydroxybutyrate. So it's just abbreviated on this side. So sorry, and that's favored due to high NADH from beta oxidation. Okay. And then acetoacetate can form acetone in non-enzymatic decarboxylation. Cool. So that's ketogenesis, and we have ketolysis as well, or ketolysis, whatever you prefer. So this happens in extrahepatic tissues, so the brain, the heart, and the muscle. The liver can't use ketones because it lacks the Scott enzyme. So that's here on our diagram. And in terms of the reasoning for this, or why we would do it, it's for using ketone bodies, especially this beta hydroxybutyrate for when ATP production from glucose is scarce. So we need another source. And the key steps are overviewed here. So beta hydroxybutyrate forms acetoacetate. Acetoacetate forms acetoacetyl-CoA, and acetoacetyl-CoA forms two acetyl-CoAs to enter the TCA cycle. So similar to fatty acid biosynthesis and fat metabolism, you can kind of see that ketogenesis and ketolysis are sort of the opposite of each other. Okay, so we absolutely whizzed through that, which is fine because there's a lot more to come. Um, this is supposed to be an interactive example MCQ question, but I need to get my notes up, so I might have to slightly squidge the screen a bit. I hope that's okay and that you guys can still see it. Otherwise, I don't have my explanations for what the right answers are. If anyone wants to pop an answer in the chat, that would be great. No pressure to though. So there's 5 questions. They're all true or false, which I should say you rarely get true or false questions in multiple choice exams, but these are taken from the MCQ bank. So they're kind of a good idea of what sorts of facts come up, but in terms of whether there's going to be a true or false coming up, I doubt it. Let me see. No one's feeling chatty today. Okay, I'll get my notes up and we can go through it instead. Okay, cool. So energy yield on complete oxidation of fatty acids is only 1/2 of what could be obtained from an equivalent weight of glucose. Is that true or false? So fatty acids yield more than double the energy per gram compared to glucose. And we did cover that in the first slide, to be fair, and quite quickly. So I'll let you guys off, but that's false. The next one about insulin promoting the uptake of fatty acids and their storage as triglycerides in adipose tissue. Have we got true? Yeah, absolutely. Well done. That's true. So, yeah, well done, guys. So, insulin activates lipoprotein lipase in capillaries, and that promotes the uptake of fatty acids into adipocytes. Also stimulates tag synthesis and inhibits hormone-sensitive lipase to prevent fat breakdown. Thanks, guys. Well done. Next one, dietary fatty acids are packaged by the liver into very low density lipoproteins for distribution to the rest of the body. So VLDLs. Yeah, perfect. Well done to those of you in the chat. That's false. So dietary fats, and it's been a bit cheeky, dietary fats are first packaged into chylomicrons by enterocytes in the intestine. And VLDLs are made by the liver. Cool, well done. Next one, glucagon promotes fatty acid biosynthesis in the liver by activation of acetyl-CoA carboxylase. Anyone want to hazard a guess at that? You've been doing great so far. Yeah, perfect. False as well. Yeah, Well done, guys. So false. Glucagon inhibits fatty acid biosynthesis. Yeah, it works by inhibiting acetyl-CoA carboxylase through phosphorylation via pKa. And fatty acid synthesis is stimulated by insulin, not glucagon. Cool. Last one. Adrenaline stimulates fatty acid mobilization from adipose tissue. Good. You guys are on fire. So true this time as well. Perfect. Adrenaline activates hormone sensitive lipase via pKa to trigger tag breakdown. And then that releases NEFAs into the circulation for use by other tissues. Cool. Okay, so that is lipid metabolism done and dusted. Let's have a glance at, well, not really a glance. Glucose is probably the biggest one, as you can tell by your syllabuses. So we'll probably go through the overview stuff relatively quickly. Um, yeah, because a lot of it's kind of common sense again. So, importance of glucose. Why is glucose a big deal? It's the body's go-to fuel, isn't it? So, especially for the brain, the red blood cells, and the renal medulla, which rely on it almost exclusively, as you could see in the fed versus fasted state table before. So, it's also super quick. Glycolysis can generate ATP even when oxygen's scarce. And beyond energy, glucose helps keep blood sugar levels in check and acts as a building block for other key molecules like glycogen, lipids, and nucleotides. And this is our overview of the pathway. So you get all of them on here for free, and you get all your definitions there for free as well. So yeah, we won't go through those just because we're going to go through them all in detail now anyway. But they're there as kind of brief reminders if you want to use them in your notes. Cool. I don't know. Are we? Oh gosh, this is terrible. There we go. That looks better, I think. Okay. Glucose uptake then. So let's kick things off with how glucose actually gets into cells. So most tissues use these glucose transporters. So they rely on facilitated diffusion, aka no energy is needed. Each of the isoforms differs in insulin sensitivity and affinity for glucose, and that's Km. I think that's on the table as well. Yep. So, for example, glucose transporter 4 is insulin dependent and found in muscle and fat, whereas glucose transporter 1 is always on and great for tissues like the brain or red blood cells that need that constant glucose supply. Cool. And then we have SGLTs, so sodium glucose link transporters, and they're in the intestines and kidneys and use a sodium gradient to actively absorb glucose against its concentration gradient. Um, cool. And then we've got a key point at the bottom. So muscles use glucose transporter 4, and that's insulin sensitive. So during exercise, even if insulin's low, glucose transporter 4 still gets to the membrane, and that's thanks to calcium. And I've kind of expanded on that. I was going to say a little bit, but this is not a little bit. I've expanded on that here. It's not the most important bit of information you need to know. So if you're revising metabolism, then kind of just learn the headline rather than all of this. But this goes into more detail about how that actually works. So calcium levels rise in muscle cells during exercise. That activates your AMPK and calcium calmodulin pathways. That causes glucose transporter 4 translocation to the membrane without needing insulin. And in terms of why that matters, it keeps glucose supply going during exercise when insulin is low. And it also helps in type 2 diabetes when insulin signaling is impaired. Um, so, yeah, calcium is doing way more than just muscle contraction and what you learn about in physiology. Okay, I hope that makes sense. Again, like I said, not the most important bit of this topic. Um, So yeah, the hormonal regulation of glucose uptake now. So insulin rules the fed state. It's released in response to high blood glucose, promotes glucose uptake into muscle and fat by increasing glucose transporter 4 at the membrane, and it encourages glycogen storage and glycolysis while inhibiting gluconeogenesis. AKA, there's no more need to make glucose when we've already got loads. Whereas glucagon looks over the fasted state. So it's released when blood glucose is low, and it does the opposite to insulin. So it stimulates glycogen breakdown and gluconeogenesis while it inhibits glycolysis and glycogen synthesis. And muscle doesn't respond to glucagon, liver, the liver does. Is that mentioned here? Yeah, it does. Okay, cool. So that's glucose uptake, and I realized that was a lot of slides with a lot of text on them. So here is a diagram that sort of overviews the hormonal regulation of glucose and the key players in that. So yeah, we've covered glucose uptake. The next thing to think about is glycolysis. So glycolysis is our starting point for breaking down glucose. We're taking one glucose molecule, so 6 carbons, and splitting it into 2 pyruvate molecules, each with 3 carbons. So in the process of doing that, we're making ATP and NADH. So it's a key energy yielding pathway. And that happens in the cytoplasm and doesn't need oxygen. So whether you're sat listening to me waffling or sprinting, glycolysis is going to help you out. And if oxygen is around, pyruvate then heads to the TCA cycle. And if not, it becomes lactate to keep things ticking over. Okay. And that's kind of the main diagram you need to know. If you have a whiteboard and whiteboard pens, I'd recommend literally just writing it out again and again until you kind of got it nailed. And that was the easiest way for me to remember glycolysis in first year. But, to each their own. Flashcards might work better for some. Okay. So we didn't really touch upon this, but the phases of glycolysis, so it's in two halves. We invest some energy and then we get a nice payoff phase. So first we burn 2 ATP to prep glucose, converting it into this fructose 1,6 bisphosphate, which locks glucose into the cell and commits it to glycolysis. Then it's the payoff phase. We get two, no, not two, yeah, 2 NADH, let's say, and 4 ATP as we turn that molecule into pyruvate. So you get the net gain of 2 ATP and 2 NADH. Right. Regulation. How do we control this pathway? There's going to be 3 key enzymes. So we've got hexokinase or glucokinase that starts the process. Hexokinase is inhibited by its product, which is G6P. Glucokinase found in the liver and pancreas, and it acts like a glucose sensor. Cool. Okay, PFK1, that's the big rate limiting step. So a super important thing that you'll probably get asked about. And that's activated when energy is low and is inhibited when we're full. Cool. And last one, pyruvate kinase finishes things off and is activated by earlier glycolysis intermediates in a nice sort of feed forward way. And hormonally, insulin basically says go, glucagon says stop. And that's via transcription for hexokinase and phosphofructokinase, and by phosphorylation for pyruvate kinase. Cool. More glycolysis. I'm just keeping an eye on the time. So yeah, in terms of pyruvate, the fate of pyruvate, that depends on oxygen. So with oxygen, it becomes acetyl-CoA for the TCA cycle. Without oxygen, it turns into lactate, regenerating NAD+, so glycolysis can keep running. And in terms of energy down here, Glycolysis nets 2 ATP and 2 NADH. Um, so once you've added in the link reaction and the TCA cycle, you're looking at a full sort of 30 to 32 ATP per glucose, depending on how NADH gets into the mitochondria. Fab. Let's zoom in a bit more on pyruvate dehydrogenase then. So the enzyme that links glycolysis to the TCA cycle, and that's where I leave you guys to go to someone else. So it's a multi-enzyme complex that converts pyruvate to acetyl-CoA, and this step is irreversible. So once it's in, there's no going back. And PDH is regulated by energy state. So when ATP is high, PDH is phosphorylated and turned off. When energy is low, it's dephosphorylated and activated. And there's a bit of clinical relevance there, um, extra. So PDH deficiency leads to lactic acidosis. Arsenic poisoning also targets this pathway. And one sign is garlic breath. So yeah, interesting to know. Okie doke, utilization of other monosaccharides. So not all sugars are created to be equal, but they all sort of find their way into glycolysis eventually. So fructose is absorbed from sucrose, then in the liver it's phosphorylated to fructose 1 phosphate and split into DHAP and G3P. And they're both glycolytic intermediates. And then in the muscle and the adipose, it's a bit simpler. Hexokinase phosphorylates fructose to fructose 6 phosphate and it jumps straight into glycolysis. And then we've got galactose. So galactose is from lactose, and that takes a few more steps. It's converted into galactose 1 phosphate, then to UDP glucose, and finally to glucose 6 phosphate via something called the Lillois pathway. Oh, also, I forgot to mention, but when you get copies of these slides, if you bring up the speaker notes on quite a few of the slides, actually, there's some part B stuff or right-hand side stuff in the notes as well. But yeah, that might be useful for some of you guys who are interested in metabolism and writing essays on it. Cool. Next up, pentose phosphate pathway. So it doesn't make ATP, but it's still important. It runs mainly in the tissues doing loads of biosynthesis. So the liver, adipose, adrenal cortex, and also it happens in red blood cells, and that's for antioxidant protection. And you can see here in this diagram, there's two stages. So we have an oxidative phase that makes NADPH, and that's really crucial for fatty acid synthesis and fighting oxidative stress. And then we have a non-oxidative phase, which is reversible, and that recycles sugars back to glycolytic intermediates like F6P and G3P, or makes ribose 5-phosphate for DNA and RNA. Okay, sorry, I was just trying to check the chat to see if there's any questions so far. Fab. Okay, fates of the products then. So basically, NADPH powers loads of biosynthesis and antioxidant processes. Ribose 5-phosphate down here is a precursor for nucleotide synthesis, so also super important. And then other carbon sugars can just loop back into glycolysis if energy is needed. So the pathway, it's basically important because it's flexible. It supports growth, it supports defense, and metabolism, depending on the cell's need. Cool. Okie doke. Next up is storage of glucose. If it comes up, yeah. So glucose is stored as glycogen, a branch polymer of alpha glucose. In the liver, glycogen helps regulate blood glucose and it lasts for about a day of fasting. Whereas in muscle, it's all about local use during exercise, during energy use, and doesn't get shared into the bloodstream. If we think about glycogenesis next, so when glucose is plentiful, we store it. Glycogenesis kicks in during the fed state, forming glycogen from UDP glucose. So glycogenin provides the initial scaffold, glycogen synthase builds the chain, and the branching enzyme makes the molecule compact and accessible. And that process is sort of illustrated in this diagram here. In terms of glycogenolysis, when energy is needed, so for fasting or exercising, we break down glycogen. Phosphorylase cleaves glucose as G1P, and when it reaches the branch, debranching enzymes are going to take over. And I guess a key point here is about the difference between muscle and the liver again. So the muscle uses it locally, whereas the liver shares it with the rest of the body. So kind of touching upon the same point as before. Okie doke. Regulation of that hormonal control again. So glucagon and adrenaline activate PKA. They promote glycogenolysis and blocks storage. Insulin does the opposite. It activates phosphoprotein phosphatase and encourages glycogenolysis. No, sorry, glycogenesis. I don't know why I said glycogenolysis. They're all too similar. It's a neat hormonal tug of war, basically, that depends on energy state. So fed versus fasted. Cool. Okay. Gluconeogenesis. This is a slide from last year, so apologies if I end up stumbling. But when glucose runs low, the liver and the kidneys, to a smaller extent, step in to make it from scratch. And that's basically what gluconeogenesis is. So it's not just glycolysis in reverse, it bypasses key irreversible enzymes and steps using alternate enzymes. And it's quite energy intensive. So it uses 6 ATP per glucose. And that's summarized in that diagram there. Okay, in terms of the substrates, we're thinking about where do the carbon backbones come from? So glycerol from fats, lactate from anaerobic glycolysis, oxaloacetate from the TCA cycle, and glucogenic amino acids. They can all funnel into this pathway at various different points to generate new glucose. And there's a bit more detail there, but I'm just conscious of time, so we're going to move on to how is futile cycling avoided? So to prevent futile cycling, basically gluconeogenesis and glycolysis are reciprocally regulated. What stimulates one inhibits another. And glucagon is a key player in that. So it strongly promotes gluconeogenesis during fasting. This is right-hand side of the syllabus, but it's important anyway. So why can't fatty acids be used to produce glucose? So when, well, so fatty acids become acetyl-CoA, and once they're in, they can't go back because pyruvate dehydrogenase, or PDH, has essentially locked the door behind them. So that's one reason. Plus, the TCA cycle burns off 2 carbons as carbon dioxide per turn. So there's no net carbon gain for glucose synthesis. Cool. So that is glucose metabolism, a whistle-stop tour. And here we have another silly true or false MCQ. So if you guys want to answer in the chat again, we'll go through the first one rather quickly. So degradation of glycogen by phosphorylase results in the release of glucose. True or false? Thanks, Leanne. Um, so, hmm, you're all going with true. And I don't want to say you're incorrect, but the mark scheme from the bank says that that's false. And I will tell you why. So, glycogen phosphorylase breaks glycogen into glucose 1 phosphate, not free glucose. In the liver, glucose 1 phosphate makes glucose 6 phosphate, which makes free glucose via glucose 6 phosphatase, which is then released into the blood. In muscle, this doesn't happen. So glucose stays phosphorylated and is used locally for energy. Yeah, maybe it's the difference between phosphorylase and phosphatase. Thanks for pointing that out. Cool. So, next one, adrenaline stimulates glycogen mobilization in both liver and skeletal muscle. True or false? Anyone fancy? Okay, here's what we'll do because there's only 9 minutes left. We'll just run through them and I will let you know that also the answers are in the speaker notes. If in doubt, answers are in the speaker notes. Oh, I've got people in the chat now. Defo true. Yeah, defo true. I agree. So adrenaline triggers glycogen breakdown in both tissues via the CAMP pathway. Um, cool. Third one, phosphorylase kinase is activated by calcium ions. True. Glycogen synthetase is activated by dephosphorylation. Also true. Muscle glycogen breakdown contributes directly to the maintenance of the blood glucose level. That is false. So let me just see the chat. Ah, okay. I don't know what's going on anymore with those answers, but they are also in the speaker notes if you want them, with justifications as to why. Right. Absolutely rapid fire. We're gonna do amino acid metabolism, and you know what? It's not actually that long anyway. And like I said, all the part B stuff or the right-hand side syllabus stuff you can find examples of at the end of these slides. So you'll get a copy of them once you filled in the feedback form. So don't worry too much about that. Right. Protein digestion. Where is it happening? It kicks off in the stomach. Hydrochloric acid from parietal cells unfolds proteins and activates pepsin. And that's basically a broad spectrum endopeptidase that starts chopping things up. Then in the small intestine, pancreatic enzymes are going to take over. So trypsin cleaves at arginine and lysine, while chymotrypsin prefers these bulky aromatics like tyrosine and phenylalanine, phenylalanine, alanine, alanine. Sorry, this is actually such a bad presentation to give as someone who can't pronounce things. Um, but yeah, they're written on the slides anyway. You'll be fine. And together, those guys break proteins into smaller peptides and free amino acids. And there's an overview of that there as well. Cool. Uptake and absorption. So at the brush border, free amino acids are taken up by sodium-linked symporters, while peptide-1 helps bring in di and tripeptides. And then basically, once they're inside, cytosolic peptidases are going to finish that job. So breaking everything down into free amino acids. And from there, they head into the hepatic portal vein to the liver. Um, I think hepatic portal vein came up as an MCQ in my year, so one to remember, guys. And fun fact, not really, but branched chain amino acids like leucine are preferentially snapped up by muscle for energy and protein synthesis. And then here is that overviewed. In terms of the sodium linking, you can kind of see this nice flow of sodium in this diagram through here. So we've got that co-transporter or symporter there. And then we've got our sodium potassium pump that helps that process out, which you guys will have learned about in physiology. Okay, overview. And then amino acids. My first ever tutorial at Oxford was about amino acids, and we had to list all of them on a whiteboard in a pair. So this gives me a little bit of trauma, but basically, you kind of just need to know the difference between essential and non-essential, um, and also conditionally essential in this case. So ones that are needed in the diet during illness or infancy. And then we've got a little bit of information here about protein turnover. So what is it? It just means constantly broken down and resynthesized. Amino acids are recycled into new proteins or used for energy metabolism. And then we've got a bit about nitrogen balance. So we can have a positive or a negative nitrogen balance. And like I said in one of my first few slides, if you guys want my essays, I actually did a terrible protein essay in first year, but it's got really good feedback that kind of clarifies some good um, or common misconceptions about protein metabolism. So if you want that, I can give it to you. Anyway, what happens to amino acids? They're fates, so they're incorporated into proteins, converted to derivatives, or used as fuel. Um, yeah, and there's an overview here of the types of amino acid. This comes up as well on multiple choices. So glucogenic versus ketogenic versus being both. Oh yeah, and here's the sort of memory tool to remember which one's which. So leucine and lysine are the only ketogenic ones. And then you can remember the ones that are both from this sort of PIT or PI triple T. Um, and then inevitably anything else that comes up on your screen when you're doing the multiple choice that isn't one of these seven is going to be only glucogenic. So just to help with memorization there. Okay, actually rapid fire this time. So amino acid metabolism. Amino acids are metabolized mainly through transamination and deamination, and nitrogen is going to be converted into urea for excretion, and carbon skeletons enter various pathways, including these four here. So, before metabolism of amino acids can continue, their amino group has to be removed, and that involves deamination, of which there's three types that I've circled. The most important one, or the one that's listed on the syllabus even. This, again, another diagram from an essay I did, so apologies for that. But oxidative is probably the most important one to get to grips with, because most amino acids are going to funnel into glutamate, which undergoes oxidative deamination. And then we have NH3 that's excreted as urea. Cool. Transamination is the moving of nitrogen basically, and we've got two diagrams there that display how that happens. So we're transferring NH2 from an amino acid to a keto acid, which is often alpha ketoglutarate, and that basically forms a new amino acid and a new keto acid and is catalyzed by aminotransferases, and requires vitamin B6 essentially. So PLP, you can see on this bottom diagram, it's a cofactor for the transaminase enzyme. And this is reversible. So it's really good for producing your non-essential amino acids that your body's supposed to be producing. But it's also good for fuel usage as well. Okay. Deamination, kind of talked about it already, but this is just some more detail in terms of the enzyme that's involved. So glutamate dehydrogenase, which is why most are funneled into glutamate dehydrogenase. Okay, we've got 1 minute left. Amino acid metabolism. Oh yeah, so urea cycle. So if we want to get rid of ammonia, then we have to use urea synthesis. And this is a really good diagram for basically showing how that happens. It takes place in the periportal hepatocytes. The first two steps are in the mitochondria, so these pink ones, and then the blue steps down here on this diagram happen in the cytosol. So basically, we're just converting toxic ammonia into non-toxic urea for safe excretion by the kidneys. And there's a cycle overview there. Okay, regulation. So CPS 1. So where's CPS 1 here? Carbamyl, phosphate synthetase 1, the rate limiting step. So this guy with the star against his name, he is very important and is activated by N-acetyl glutamate. Okay, so enzyme activity increases when amino acids break down and NH4 levels rise. And then there's some info on chronic regulation there, which is less important to know for your part A's. Okay, that diagram's also a really nice summary. Oh, and then there's a tiny part on the left-hand side of the syllabus about amino acid metabolism. So I've written a little bit there about muscle. If you want the right-hand side stuff about the other sites that I mentioned, that's all in your speaker notes as well. But this is a really tiny part of the syllabus. Even the Carhill cycle is on the right-hand side. That's also got some notes down here in speaker notes. I'm just conscious that we're at the end of the session now, and I don't want to keep you guys. But yeah, so again, an example MCQ with the answers there. And thank you for listening. And you can see I actually do have extra slides for you guys. I promise I wasn't just lying. So you can complete the feedback form to receive a copy of the slides and email me for those essays if you want to. I'm not forcing them onto you, I promise. It sounds like it, but I'm not.